Good morning, students. Today we are going to study about cell division and all those topics that are related to cell division. That's definition, types of divisions, differences between them, cell cycle and mitotic cell division. Now the definition of cell division is it's the phenomenon of production of daughter cells from the parent cells. The types of cell division where the daughter cells are formed. There are two types in a sexually reproducing organisms that we are dealing with today, mitosis and meiosis. So mitosis is a type of cell division in which the nucleus divides to produce two daughter cells, each with the same number of chromosomes as the parent cell. But in meiosis, in this type of cell division, the chromosome number is reduced to half, which is also known as reduction division, and this results in the formation of four haploid cells or chromosomes having only one set of chromosomes in the daughter cells, and it results in the formation of gametes. Now we come to the differences, the specific differences sometimes are asked between the mitotic and meiotic cell division. Mitosis occurs in the somatic or body cells, meiosis in the reproductive cells of gonads or meiocytes. And mitosis occurs continuously throughout life, but meiosis only occurs in the reproductive age. And mitosis provides growth, repair, and replacement. This is its significance, but meiosis only is responsible for gamete formation. The two daughter cells are produced at the end of mitotic division, whereas in meiosis, four daughter cells are produced. The full set of chromosomes are passed on to the daughter cells in mitosis, so that cells are diploid or having two sets of chromosomes with 2n number. Whereas in meiosis, the daughter cells contain half the number of chromosomes, that is, into each daughter cells, or that is haploid set of chromosomes, or only one set of chromosome is passed on to the daughter cells. Now, the number of nuclear division that takes place is after the chromosome duplicates is a single division, whereas two nuclear division occurs in meiosis. The chromosomes are identical, the genes and genetic constitution in mitosis, whereas the genetic constitution in the daughter cells are different from the parent cells, and the chromosomes and the genes are randomly assorted in the daughter cells, which results in genetic variation. Now this is cell cycle. The cell cycle is a series of events that take place in a cell before it prepares to divide again, which includes two periods, that is the growth period of the cell and the division of the cell. So the cell cycle defines it to be the period from one division of a cell to the next cell division. And the growth period in which the cell prepares itself is known as interphase, and the other phase where the cell actually divides to produce the daughter cells are the M phase of mitosis or mitotic division. So in the interphase, what happens? It is the period of growth between the two successive division of a cell, and in this period the cell grows in size and attains the parent size, cell size and shape, and also the chromosomal matter, the cytoplasmic matter also gets doubled. So this interphase involves three stages, G1, S and G2 phase. These three stages, what are the changes that occur during G1 phase? In G1 phase actually, where the RNA and proteins are synthesized, the cell organelles that contain DNA like mitochondria and chloroplast, they replicate or produce more of their own kind, and the cytoplasmic matter doubles or increases in size so that the cell can divide into two equal sized cells, right? So this is where the cell contains in the G1 phase is completed. At the end of G1 phase, the cell has two options, where it may enter into S phase or it may leave G1 phase and enter into G0 phase. This G0 phase is the resting phase where it escapes the cell cycle and stops dividing temporarily or permanently. Temporarily in the case of liver cells, skin cells, right? And in permanently in the brain cells or nerve cells, where the new cells are never synthesized. But if there is uncontrolled non-stop cell cycle, it may lead to the formation of tumors which can be benign or malignant. But once the cell enters into S phase, it cannot revert back or stop dividing. It must enter into M phase. So in the S stage, what happens? The chromosomal matter increases by the duplication of the DNA. So the DNA replication occurs in S phase, DNA replication. So more of RNA are synthesized, more of DNA molecules that require to be synthesized like protein and nucleotides are synthesized. So the chromosome starts to duplicate. And in the G2 phase, that is in the phase more RNA or proteins that are required to be synthesized are synthesized, and the cell is now ready to divide, that is in G2 phase. And then after the G2 phase is completed, which is a very short period, it enters into M phase. Now the M phase or mitotic division phase involves two steps, that is karyokinesis and cytokinesis. And karyokinesis occurs in four phases: prophase, metaphase, anaphase, and telophase. And at the end of the karyokinesis, the cytoplasm divides, which is known as cytokinesis. So the division of cytoplasm occurs, which finally produces two daughter cells. Now we come to the M phase or mitotic phase. In the mitotic phase, this is the shape and the structure of the interphase stage. Prior to mitotic division, the chromatin matters as seen, chromatin threads, the intact nuclear membrane, the nucleolus, and centrosome centriole complex. The chromosome, which was single strand, now starts to divide or replicate to form two daughter chromosomes. Each chromosome forms two daughter chromosomes. Next we come to karyokinesis or the nuclear division, which involves the first phase, that is prophase. Now, in prophase, I am showing you two cells, animal cells and plant cells, and here we will see the differences between them. In animal cell, what are the events that occur? The identifying feature, you can say, of prophase. You can see the centrosome centriole complex splits into two in pairs. So, centrioles in pairs are separated or splitted to form two centrosome centriole complex. The chromosomes, as you can see here, become very distinct. Duplicated chromosomes are observed with their paired chromatid, sister chromatids, right? And then we find here from the centrosome centriole complex these radiating fibers, which are called as astral rays or astres radiating, which extends to form the spindle fibers with the addition of more of microtubules. These microtubules are present in the centriole of the centrosome. In the hollow cavity of the centrioles contain the microtubules which attaches themselves to the astres, and that leads to the formation of the spindle fibers. The nuclear membrane and the nucleolus starts disintegrating. It starts disintegrating and enters. Now, they get merged with the cytoplasm. Similarly, in the plant cell, only difference you find here, there is no centrosome centriole complex, but the chromosomes have duplicated. You can see the nuclear membrane and the nucleolus is starting to disintegrate, and finally it merges with the cytoplasm. But even in the absence of centrosome centriole complex, with the absence of astres, they still form the spindle fibers with the addition of microtubules which are located below the cell membrane. Now, after this, you have observed the identifying feature of the prophase. We now come to metaphase. In the next phase, the metaphase, you can find these duplicated chromosomes have oriented themselves and arranged themselves on the equator. And as they are lined up or in a linear fashion, all the four chromosomes are arranged. So you can see here, and in the plant cell also, you can find the duplicated chromosomes are arranged on the equatorial plane. And the centrosome centriole complex that's completely at the two polar ends, right? The daughter centrosome complex. And from the astres in the animal cell, you can see the spindle fibers are extended from one end to the other, forming a spindle-like structure, and the spindle fibers remain attached to the centromere. So these centromeres perform two functions. They attach the chromatids and also allow the chromosomes to be attached to the spindle fiber, as a result of which they help in the orientation. But you do not find the nuclear membrane here. In the similar way, in the plant cell, the chromosomes are arranged without the nuclear membrane and the nucleolus. There is no sign of them, but the spindle fibers are formed even with the absence of the asters. Now, this is the identifying feature of metaphase, the linear arrangement of the duplicated chromosomes on the equatorial plane or metaphasic plate, where the chromosomes are very distinct and they can be counted. The numbers can be counted easily. Now we come to the anaphase phase. Now in the anaphase, what do you find here? You find that the chromosomes are separated and each of the chromatids are moving away from each other towards the respective polar ends. Now here what happens? What are the events that take place here? The centromere splits and divides the chromatids into two. They get segregated, and due to the contraction of the spindle fibers, the chromosomes are pulled apart. Now these are the events that help in the movement of the chromatids away from each other. And this movement of away of the chromatids from each other, of the daughter chromosomes, identifies anaphase. And similarly, you can find this also in the plant cell. And the chromatids move towards the polar ends, and that identifies them to be the telophase phase. Now in the telophase, what happens? Those chromatids, the daughter chromatids that were moving towards the poles, are now decondensed. They become long and thread-like and are now called as chromosomes, not as daughter chromosomes or daughter chromatids. Now you can see them on the two ends, and there is a new set of nuclear membrane being formed around each set. In both plant and animal cells, you can observe this. And the two nuclei are formed. This identifies the telophase. But the spindle fibers are contracting, and finally they form the asters in the animal cell. Towards the end of the telophase, you can see that there is a constriction in the animal cell, the cytoplasmic constriction occurring from the cell membrane towards the center. And as this furrow that is formed, it deepens and separates the cell into two cells. So these are the two daughter cells, and here because the cytokinesis, this is called a cytokinesis, which is extending from the periphery towards the center. That's why it is called as centripetal cytokinesis. But whereas in case of the plant cell, there is rigid cell wall, so a cell plate forms in the center between the two sets of nuclei by the deposition of vesicles of the Golgi bodies. And this cell plate extends from the center towards the periphery, which divides the cell into two daughter cells. Now this is known as centrifugal cytokinesis. Now this ends the division of the mitotic division, both plant cell and animal cell. I suppose you must have been able to identify the features in each phase of the chromosomal behavior. Now this is a video to show the different stages of mitotic cell division. I want you to identify the stages from this video. This will help you in future how the chromosome behaves in different phases till the end. Thank you.